Hi everyone, welcome back. Starting up the AMP2 stuff now. So what we're going to be working through is really talking about kind of finishing up the nervous system. This is going to be a combination of doing the central nervous system as well as autonomics. From there we'll start working through a lot of the rest of the other systems until we finish everything up. So first thing we're going to be looking at here is the idea about the central nervous system. It was always back in the day, it seemed like Aristotle, who was the picture of this one right here, kind of the main biologist of the day, thought the heart was what was in charge of everything. Obviously, we know that's not particularly the case, but at least he was looking into stuff like this. So, what we know is that the brain is really the particular structure that is doing a lot of kind of determining who we are, how our body works, doing a lot of the command and control within the body. It's housed inside the skull, inside the cranium. About 1,500 grams on average. 10 to the 12th neurons. So you're looking at kind of 100 billion neurons. With some of those having, at least in the cerebellum, up to 200,000 synapses, which means connections between these. So again, lots of interconnectivity, lots of integration in this one. Again, really what makes us us, and to me this is always one that I always think is kind of interesting. I mean, when you really think about what the brain is doing, it is incredibly powerful in terms of the amount of memory storage we have, the amount of processing we're capable of doing. I always use the example of like throwing a ball to somebody. When you're doing that one, if somebody throws a ball at you and you catch it, you're basically in your mind have to calculate where the ball is, where it's going to end up, and then being able to control with coordination those muscles necessary to move your hand to where that ball is going to end up. Again, you don't see a lot of robots or other stuff like that that are catching things like that. So, I mean, it is a ton of processing that needs to happen quickly. And again, that's on top of everything else the brain is doing at any particular time, which is kind of monitoring most of the homeostatic mechanisms in the body, making sure all that is where it needs to be as well. Again, this chapter, again, is going to be the central nervous system. We're going to be talking a lot about the brain and spinal cord. One of the things I want to say in this one, we did anatomy with the general nervous system, like how nerves work. This is a lot more of anatomy, so brain, parts of the brain, what they're responsible for. We're going to see there's not a lot of deep physiology in this chapter, but there is a lot of knowing what part does what. So like I said, the brain is a little bit under, it's about, like I said, about 1,500 grams or about 1.5 kilograms, which is not much. Outer surface, we're going to see our brain is very folded. This is not true of all brains. That is a lot about getting surface area and being able to pack more neurons into that space, which is going to allow us to have more processing power. We're going to see that there's 4 main regions to the brain. A lot of times when you see a picture of a brain, a human brain especially, you're going to see that big kind of wrinkled surface there. Mainly what you're seeing is the cerebrum. That is what most people are used to seeing. The diencephalon, if you've ever seen a brain or an image of a brain, generally you don't see the diencephalon because that is completely interior. And then the brainstem is kind of what's going down and becomes the spinal cord, and the cerebellum is the part underneath that. And we're going to see, at least in parts of our peripheral nervous system, the cranial nerves, their part of the peripheral nervous system is going to exit from the brainstem. So this shows you these different parts. So 4 major portions of the brain. You can see the cerebrum is the main obvious part here. Cerebellum, the second biggest part. As I was kind of saying, the diencephalon is mainly interior to this, and then you have the brainstem, which is the combination of these 3 structures over on the right side of the screen here. We're going to see, again, we have lots of folds on this one. We call these folds gyri. That is the plural. Gyrus is singular. Those depressions or deeper dives into it are going to be sulci. Singular would be sulcus. And then we're going to see there is certain areas where these are much deeper grooves or fissures in here. We call these fissures. So a deeper, kind of to me, it's like a canyon versus a kind of a shallow depression. So those fissures a lot of times are going to be what separate the cerebral hemispheres. So median longitudinal fissure, transverse fissure separating the cerebrum from the cerebellum. And a lot of times when we're talking about this, we can talk about more towards the nose or anterior side. That would be rostral versus caudal. Sometimes terms that are used with the brain. So again, those raised areas are those gyri. The deeper valleys are the sulci. We're going to see there is a lot of times when we get into the brain a little bit more, we're going to see there is of the cerebrum multiple lobes to this. A lot of times these are defined by slightly deeper sulci, things like the central sulcus, the temporal sulcus. Those are going to be some of these deeper, more prominent sulci that separate that cerebral cortex into its main areas. So first thing we're going to get into is really how is the nervous system organized, and then again, what is the role of the brain within the nervous system? Again, what parts are doing what mainly? So the first question here is really how is it organized? So one of the first things I want to talk about is the protective layers of this. This is going to be there's a lot of parts to the brain. And honestly, the brain is probably one of these areas in the nervous system, especially the central nervous system in general, is probably one of these things that 20 years from now, this is one of the chapters that maybe have a lot more in it or more knowledge in it. There's still a lot in the brain that we don't know exactly how it works. I mean, for example, like the drugs that knock you out for surgery, we know they work, but we don't know what the actual mechanism is that actually renders you unaware and able to perceive. We do know that they work though. So again, we don't know everything within the brain. So this is an area, like I said, there may be a little more. The protective layers that we're talking about, these are referred to as meninges. If you've ever heard of meningitis, it is an inflammation of these. There's multiple protective layers that are going to cover the central nervous system, protect it, and surround the blood vessels, really protecting things that are dangerous, ideally from getting into the brain. This is going to make the cerebrospinal fluid is going to be housed within these ones, help protect through really cushioning and shock absorption, that brain. We're going to see they have 3 layers to the meninges that we refer to. These are all different types of what they call mater. You have the dura mater, which is on the outside, the arachnoid mater, gets its name because it looks like a spider web, on the middle layer, and then the really fine layer is that pia mater, which is the most interior, closest to the brain surface. And this is again kind of showing you a skull kind of dissected and showing you the different layers. So you can see the skin, some of the connective tissues, the bone, and then you can see this dura mater, arachnoid mater, and you can see each of these are covering the surface of that, not maybe going into every nook and cranny. They don't say pia mater on here, but that is a very fine layer that is going over every gyrus and sulcus, that it's going all down and around everything. The dura mater we're going to talk about in just a second. It is incredibly tough. Generally, when we can do this in person, we will actually dissect sheep brains that have the dura mater intact, and it is quite tough and leathery. You have a very difficult time trying to take this apart or tear it. To me, it reminds me of a very strong nylon. Like if you think about like a tarp or something else like that, it's strong like that. So like I said, the dura mater is a lot of dense, irregular connective tissue built into this. It is the most superficial layer. Like I said, covers everything. There's certain areas where it folds and has a fluid-filled space within it called a dural sinus. If you've ever heard of a subdural hematoma, maybe at some point in your life, that is a bleed underneath this dura mater, which a lot of times is going to hold that pressure in, and that's why a lot of times they will have to open up the skull to relieve pressure when there is some type of bleed within the brain, because any pressure on the nervous tissue tends to stop its functioning. Again, they talk about a couple main layers to this. So the layer that is closer to the arachnoid mater, they call the meningeal layer. The layer, the upper, more outer part of this dura mater is called the periosteal layer, which is in contact with the inside of the skull. These are fused together really well. They do have some blood-filled spaces in here, these venous sinuses in here. A lot of times you'll see it on a cross-section of the brain. It's triangular in nature in the center there. Helps to drain some of the blood out of the brain. We will talk about the blood-brain barrier later on. Blood still does go to the brain. It's just on that one, as we've said before, a lot of layers of cells, extra cells surrounding that really limit what gets in or out. Underneath this is what they call that epidural space between the dura mater and the skull. That's that epidural space. Sometimes they can provide drugs into these areas if need be. The arachnoid mater is underneath that. It looks kind of spider webby. It gets its name from that. The space underneath this is filled with, it has blood vessels traveling through it, and it's filled, and the space is occupied by cerebrospinal fluid. That's what the CSF stands for in this slide. Again, helps to cushion the brain and that. The pia mater is the innermost layer. It is really just areolar connective tissue. So that really fine connective tissue, the loose connective tissue connected to the brain, follows every contour of that. Again, meningitis is inflammation of these ones. It can be caused by viruses. It can be caused by bacterial nature. Again, both of these can be quite dangerous to people if not treated, or if pressure gets to build too much, there's too much inflammation. Both viral as well as bacterial meningitis can end up being fatal. Again, that arachnoid mater has that subdural space, which is housing. It's that space underneath the above the arachnoid mater, but beneath the dura mater. If fluid gets in there, that's where you would see that subdural hematoma, for example. And again, this is just another cartoon version of it, showing you these different layers and where they're at, kind of. And to me, again, important to know what's the outermost, what's the middle layer, what's the innermost layer. Again, there's going to be a lot in these chapters. This is not going to be a write me an essay on all the different parts of the meninges, but you should understand what the layers are, which order they're in, and again, some basics of what they're made of. Cerebrospinal fluid is this fluid that is underneath the arachnoid mater, in between that and the pia mater, in that subarachnoid space. It is made within the third ventricle of the brain, which we'll look at the ventricles in just a second. These are fluid-filled spaces within the brain. It is a blood filtrate made at an area called the choroid plexus. This is an area where that blood-brain barrier is not complete. We can make a blood filtrate, and that is what is going to become the cerebrospinal fluid. It is taken back into different kind of capillary networks surrounding the brain and spinal cord. That is going to help reabsorb this. We are generally making more cerebrospinal fluid in any particular day than is what is in there at any time. So what this is meaning is it does have to make its way from where it's being made to where it is reabsorbed. If that was not to happen, or there was some type of blockage, that fluid could build up and cause problems. Again, it provides shock absorption, helps support the weight of the brain so it's not pushing down on the bottom of the opening of the skull, and it's also going to provide a lot of nutrients to a lot of these nerve cells. So this is kind of showing you, obviously, in a dissected brain, you will not see fluid in these spaces because, again, if you cut it open, the fluid's going to drain out. But you can see in each of these ones, this is that little weird capillary network, that choroid plexus in these third ventricles that is going to make that blood filtrate. It's going to fill these spaces as well as go into other parts of the ventricles within the brain. So you can see it right here. Again, this reduces the weight of the brain. The brain is mainly lipid tissue. It is going to float somewhat on the cerebrospinal fluid, which is mainly water. Reduces the weight of the brain on the base of the skull by about 95%. So it is really helping to protect that weight of the brain from causing any damage to it. It's also surrounding the brain entirely, which means it can also provide shock absorption for that brain moving around within the skull. If you've ever been on the highway and you've seen those barrels that are on exit ramps, a lot of times those are filled with water to kind of do the same thing, to be a shock absorber. If someone was to be driving there and instead of having them hit something super hard, that will slow it down a little bit over a little bit more time, which is more protective. So again, shock absorption reduces the weight, and again, providing nutrients in the form of getting rid of the waste, taking the nutrients in, as well as any type of hormones or other stuff like that that need to get to these cells. Again, all these ventricles that are filled with cerebrospinal fluid are lined with those ependymal cells, which had those cilia on them, which are going to help move this around. So there is a current of cerebrospinal fluid that is, again, being made in one particular area, and the flow is going to be going from that area and traveling in a defined direction, and sooner or later being reabsorbed. As I was kind of saying, we house about 150 mils of cerebrospinal fluid at any one time. And any particular day, we're making about 500 mils of this, so half a liter. So as I was kind of saying, this is continually being reabsorbed. If something was to block it, and this is where it can become an issue, is you have this aqueduct right here. All the cerebrospinal fluid is being made in here. If there's somehow damage to this, there's no way for this to get out, and you can get issues with basically these ventricles filling up like balloons and starting to press against the nervous tissue, which can be a problem. Sometimes this happens having what's the word I'm looking for? Congenital kind of defects within the brain every once in a while when somebody's born, and you can have an issue with a blockage in here, and there's something called hydrocephaly, which could be a blockage in some of the anatomy of this, which leads to a buildup of cerebrospinal fluid in here. Again, nowadays they can do surgery and other things like that to remedy this. It was one of those that used to be fatal by about 9 months in infants. Again, it's being moved around here. It flows into these kind of specialized capillaries called arachnoid villi, and is going to drain into, as well, some of these dural sinuses, which are going to let this get back into the bloodstream. So again, there's a one-way flow from where it's being made to where it's being reabsorbed. You can see how it's traveling past some of these areas, those arachnoid villi. The ventricles, again, are these fluid-filled spaces, part of how we develop. They're lined with ependymal cells and filled with cerebrospinal fluid, as well as that fluid traveling down out of the brain and down the central canal of the spinal cord. So you can see some of these in the pictures here. Again, you don't see them particularly as well. You can see them a little bit right here and here, as well as right there and there. Again, become more prominent if you were to look at something that wasn't dissected or that still the fluid in there. They generally are taking up a little bit more space. You can see this on a CAT scan, an MRI. If the baby hasn't had a soft spot yet, one of my kids had to get tested for hydrocephaly. When they saw the soft spot, they actually did a ultrasound of that, and I was actually able to see the ventricles and could see that they were normal. Like I said, you can see these on some of these different MRIs and stuff. Again, there is a number of different ventricles. You have two lateral ones here that are off to the side, as well as a third ventricle, and then they have one down in front of the cerebellum that is the fourth ventricle. I mean, to me, know that there's different levels to this, and there's different fluid-filled spaces in here. This is probably not something I'm going to be showing you images of and saying, is this the third ventricle, one of the lateral ventricles? But what I'd say is know that these things exist and a basic understanding of them. Again, I'm probably not going to be giving you any type of image and saying identify these, but I want you to understand that they're there and where they're at. So the third ventricle, like I said, in the center of the brain, the diencephalon. Fourth ventricle is another, it's much smaller than the lateral ventricles or even the third ventricle, but that is in between the pons and the cerebellum, which is connected by that cerebral aqueduct. Sooner or later, once it gets past here, that's going to merge into that central canal that becomes the part of the spinal cord. And you can kind of see all this right here. You can see the lateral ventricles, that third ventricle in the center, fourth ventricle right down here. Again, not going to give you this image and ask you to be able to label these on a quiz question or something else like that. I don't think I'm going to go there. There's a lot of other stuff that's, to me, a little bit more key. But again, I want you to know that they're there. So one of the last things that we're going to talk about on this first one here is a little bit about the blood-brain barrier. Nervous tissue is incredibly fragile, probably, I would say, relative to a lot of the other ones here. There's a lot of things that can influence its function. So like we said, drugs, waste products, a lot of other substances. We have what we call the blood-brain barrier. All these blood vessels that are going up and supplying the brain, they are going to have another layer of cells that are really serving to act as a filter and really limit what can get into or out of the brain. So certain blood cells can't travel into the brain. That's why they have to have those microglial cells that we talked about last semester. There's going to be a lot of drugs that, certain chemos for, like, example, like a brain tumor. You can do a lot of different chemos, but a lot of chemos won't actually get to the brain. So this blood-brain barrier really limits what can get in there as a way to protect. Again, sometimes that can be a detriment, but to protect what is going on within that nervous tissue. These are really parts of those astrocytes that we talked about before, making those little paravascular feet that really almost fit together like puzzle pieces. That, along with these endothelial cells, are what this blood-brain barrier is. Again, continuous basement membrane, those cells all acting, those paravascular feet are the astrocytes. These are really acting to limit what can get in or out of the brain in terms of substances. Certain things obviously can travel through this. Things that are lipid-based tend to go through a bit more. So things like alcohol can get across there, nicotine, certain drugs can get across there, but a lot of things can't. There are certain areas where it isn't present. So in order to make that blood filtrate, the choroid plexus does not have that. Any areas where we need to have the bloodstream interact hormonally and have nervous tissue access that bloodstream, we are going to have this reduced or absent in these areas. So the hypothalamus as well as the pineal gland are both things that are going to release hormones into the bloodstream. In order to do that, they need that ability to have a reduced or absent area of the blood-brain barrier in those spots. And again, this is showing those paravascular feet, and this is what I was kind of saying. It creates another highly selective membrane that anything has to travel through in order to get to that nervous tissue. And I did mention hydrocephaly. That is that condition where that cerebrospinal fluid builds up. Generally was fatal by about 9 months. It can be surgically treated now. Hydrocephalus just means water in the brain or water on the head, because cephalus is head, cephalic. Hydro is water, so it's water filling the head. If you were to see an image of somebody that had this as a child, before those skull bones fuse together, if you have a lot of excess pressure, it will push out on it a little bit more and will enlarge their cranium. Again, generally it gets tracked and found and tends not to be an issue as much anymore. Again, before this, or in countries where they can't do surgery on this, it generally would be fatal by about 9 months, because at that point the skull is starting to really fuse together a lot more and there's no way for that pressure to be relieved, and you get enough pressure on the nervous tissue, it will stop functioning. So this is our first one here. What we're going to be doing from here on out, next thing here, we'll be taking a look at the spinal cord on the next episode here, and then start working through the brain and the different parts of the brain. So I will see you next time.